within, thank you, Janet, for remembering. We can turn around the climate existential crisis within a decade or two, just by putting the carbon back in the ground. So that's what I'm going to explain to you tonight. So we have a few important quotes here. Paul Harvey, describing man, that really we owe our entire existence to the top 6 inches of soil and the fact that it rains. That a life is coming that feeds us is coming from there, or life that's feeding the animals that we eat is coming from plants. And then, when Franklin Roosevelt was president, we had the Dust Bowl in Kansas and the Midwest and the massive drought. And one of the Congress people made his point by opening the curtain in the halls of Congress and just saying, "I give you Kansas," because the dust had literally blown across the country, in massive amounts, and was very visible in Washington, D.C. And so Roosevelt understood that we are dependent on soil. And then the other thing I'm going to talk about a little bit tonight is the carbon cycle, because it is one of the major ecological cycles, in addition to water cycle and nitrogen cycle and hydrogen cycle, that keeps life churning on the planet. So you'll get to learn a lot more about that. And the 2% solution refers to that we only need to add back 2% of the carbon that's missing from our soil in order to get back to 350 parts per million in the atmosphere. That's a pretty doable thing. And it's a very hopeful thing. So we have to really start with how life, can I get rid of, are you guys seeing this screen sharing thing on the, or is it just me? Trying to get this thing out of here. We really have to start with life on the planet as how it exists, right? So we have the sun, we have carbon dioxide in the atmosphere, we have water, and in the presence of chlorophyll molecules in plants, we have the process of photosynthesis. One of the terminology misuses that's going on is the word, we always get these fad words that pop into our iconography, and that is, the word sustainable is one of them, but the word now sequester carbon. I'm sure all of you have heard the phrase sequester carbon, and people use it very loosely, but in reality, only plants can sequester carbon. So only plants can take carbon dioxide and incorporate it into their physical selves and churn it around and make a glucose molecule. So plants are what are sequestering, and they can sequester in their stems and leaves and flowers and roots. And it's everything else is storing the carbon. Okay, so the soil and the fungi in the soil and the bacteria in the soil, and the microbes in the soil are storing the carbon, not sequestering it. Okay, so that's just a really important point to get straight so we don't misuse that word. So composition on the planet is photosynthesis, and decomposition is the exact opposite, and it's done by fungi. So it's those two processes in balance, the same exact chemical formula, 6 carbon dioxides and 6 waters in the presence of sun and chlorophyll make 1 sugar, 1 glucose molecule, and oxygen as a byproduct. It's a waste product, actually. And decomposition is the exact opposite. Glucose in the presence of oxygen in an aerobic environment break the carbon bonds in the glucose molecule and return it back to carbon dioxide and water. So these two processes go hand in hand. Just a little bit about the decomposition process. There are different groups of fungi that decompose different sets of molecules in plants. So, plant skeleton, if you will, is the cellulose. So they have
We lost sound. She's not on.
Okay, is it better now? Can you hear me now? Okay, I don't know why that happened. Sorry. So cellulose is a primary cell wall, and lignin gets laid down to the inside of the cellulose wall in secondary growth. So if a plant is going to be more than an annual plant, if it's going to stick around for a biennial, 2 seasons, or a perennial, multiple seasons, the lignin will be added to the inside of the cell walls. It's also added to certain cell types in the plant, namely the vascular tissue and a little bit of the epidermal fibers that help with the structure of plants. So these different groups of fungi decompose the different parts of the cell wall. So brown rot fungi decompose the white cellulose and leave the dark brown lignin behind. And you've all seen these brown rots, here in the woods as logs rotting, and then they have this brown cubicle rot pattern to them. And then white rot fungi at the bottom do the opposite. So they decompose the lignin, which is the harder molecule to decompose. It's far more complex a molecule than cellulose, which is fairly linear carbon chain. And they leave the cellulose behind. And then those in the middle are the soft rots, which are decomposing the hemicelluloses, so smaller, less branched molecules of cellulose. So we rely on all these different fungi to convert our organic matter back into the elements, into nutrients, right? So we're taking all the nutrients and making them available again for uptake by plants, which is what decomposition does. So there are different nutritional groups of fungi. And so fungi can't make their own carbon from the sun like plants do. So they need to get their carbon from somewhere else. So they can either get dead organic carbon, which we just discussed, by decomposing, or they can kill for it. So they can parasitize a host. If you have ringworm in your toes, you are the host for Taenia fungi. And they can be a pathogen and not kill their host, but live off of it. And then they can share their carbon. And it turns out there's way more sharing going on than we previously realized, like way more. To where my whole view of life on the planet is no longer Charles Darwin's view. It is more Lynn Margulis's symbiont, endosymbiont theory. So the way fungi have done this is through forming symbioses with algae, which was some of the early primitive colonizers of bare mineral soils, so volcanic soils or other erosive bare soils, in a form of a lichen. And then it turns out we thought that was a pretty much a two-partner dance, but it turns out there's often a third partner in there, either a bacterium or a cyanobacterium. So it's now looking at tripartite symbioses with lichens. And then we have mycorrhizae, myco meaning fungus, rhiza meaning root, and those are the symbioses that are underground, unseen to us, where the fungus is either sheathing the short roots of the plant, usually a woody plant, or is actually penetrating the roots of an herbaceous plant and sharing the nutrients and water in the soil, making it far more available by way increasing the surface area for absorption, and then the plant feeding its transport sugar, which is sucrose, to the mycorrhizal fungi. And then the last one, the endophytic fungi, this is relatively new in the last 20, 25 years. I'm sure you find publications before that, but in terms of public and graduate students working on endophytic fungi, it's a couple decades old. And when we look, we're finding fungi in the cells of the leaves, in the cells of the stems. If you know the chemotherapeutic drug Taxol, that was first synthesized from the bark of the Pacific yew tree, which is a very small population in Northern California, Southern Oregon, not sustainable harvesting at all, to where microbiologists discovered the fungus in the bark of the yew tree. That was the precursor molecule for Taxol. So they're now synthesizing it from the fungus. And then groups of students from Yale have gone down looking for plastic-eating fungi in plants and have found a few by looking at leaf, culturing fungi from leaves. The coolest part of this whole climate story, one of the coolest parts, is that I've learned by studying it the last 6 years, is that the fungal cells and the bacterial cells living in the leaves of plants are actually transpired through the stomates, the breathing holes in leaves, through transpiration into the atmosphere, and they become the predominant nucleus for a raindrop to form. So raindrops can form on salt crystals over the ocean, they can form on ice crystals high up in the atmosphere, and they can form on bacterial or fungal spore particles. And that this releasing of bacteria into the atmosphere and in our vegetated tropics, it is primary rain formation catalyst. Okay, so you get the idea that fungi are important everywhere, I hope by now. And then we look at the global carbon cycle, which is way off balance right now. And I just have a couple numbers here. I'm not going to dwell on these numbers, but I want you to look at, okay, so GT stands for gigatons, and a gigaton is 1 billion metric tons, or 10 to the 15th grams. And so the atmosphere is holding 750 gigatons of carbon. The terrestrial vegetation above ground is holding 610 gigatons of carbon. And then we go down into the soil, and 1580 is more than this 1360 that we get by adding the atmosphere and the terrestrial. So the carbon soil carbon is supposed to be greater than what we have above ground vegetation and what we have in the atmosphere. And right now we are off balance with too much in the atmosphere from burning fossil fuels, but more importantly, from our agricultural practices. So when we render soils bare, we let all that microbial life die, and all that carbon dioxide, instead of being released very slowly through decomposition, gets released very, very quickly. So we're just off balance. And that is the role that the fungi play here in holding on to this carbon is really important, carbon in the soil. Okay, this is just showing the carbon cycle and just that it's way off balance. Don't need to dwell on that. Okay, so a lot of you are gardeners and farmers, and we know that mulch is a good thing. So adding dead organic matter to the top of our gardens, our fields, is a good thing, right? Because it's something to, one, keep the ground covered so that there's less heating of the soil, there's less water loss in the soil, and that it's food for microbes to decompose. But in comparison to just mulching versus using healthy soil principles where we don't till, we plant diverse cover crops, and we bring in controlled grazing by animals, and we keep living plants and living roots in the soil as long as possible, we have a much greater success story here in terms of soil biodiversity and holding on to soil carbon. So much so that, when we just mulch, we have still a 60 to 80% carbon dioxide loss, because a lot of that decomposition is happening right at the surface, right with our mulch. And if we do it by putting it through the plant roots, feeding the microbes through the plant roots, we have only a 20 to 30% carbon dioxide loss by comparison. Likewise, we only form 5 to 10% of humus in this first situation, and more like 40 to 70% organic matter in the latter situation. One statistic that I learned recently that blew my mind, because I have a botany degree, is that the photosynthetic products that 70 or 40% of the photosynthates, the sugars the plants make through photosynthesis, are transported to the root system. So what I want us to think about is that here we have these big, beautiful trees above ground. There is as much tree below ground as there is above ground, right? The root system of that tree is equal and opposite in pattern below the ground. And just because we don't see it doesn't mean that it's not super, super important. So think of the tree, think of the ground level as the middle of the seesaw, right? And it's got equal biomass above and below. So 40% of the sugars it sends to feed these very important mycorrhizal fungi and some bacteria. This is really a cool slide, but it's a slide to digest. Could be a whole lecture on just a slide. Anyway, this is the history of the earth, right? So here we have 4.5 billion years ago, and water coming on at 4.2 billion years ago, and then some bacteria, still no oxygen. So we have anoxygenic photosynthesis going on with sulfur, hydrogen sulfide. Then we get some viruses, then we get some nitrogen fixing bacteria, and then we get oxygen because of the nitrogen fixing bacteria producing some and releasing some, and then some viruses, and then fungi come on the scene, like 2.2 billion. So like half the Earth's history, the fungi have been around. And here's us at 1 second before midnight on this clock. Like we are so insignificant. And all of this life that has evolved ahead of us has, I feel, just so much information held within it. And we haven't even begun to understand. I mean, here we have the soil beneath our feet, and we know more about space than we do about what's under our feet. And that's pretty troubling. So we have this whole new frontier beneath our feet to really understand. And it's still not easy because it's all microscopic. But anyway, the point of the slide is to just show that fungi have been around for a long time. And it's this group in yellow up here, the glomeromycota, around for 500 million years, that are the soil formers. Okay, so we're going to spend some time on them. Here's a fossil evidence of their presence 440 million years ago in some chert. This fungus named fossil fungus Tortubus protuberans, and you see here the long fungal hyphal cells, a few little clamp connections even, and then you've got these large spores forming, or sometimes they're vesicles. So these are probably spores because they're thick-walled. Okay, so there's some fossil evidence already of these glomeromycota. And let me know if I'm going too fast, because I get excited and I go fast. So if there are any hands, Janet, I'm just going to pause to see if there's any questions yet.
If you do have a question, you can unmute yourself.
Okay.
Looks like maybe we can keep going.
Okay, great. Don't hesitate. So endomycorrhizae versus ectomycorrhizae. So endo means inside. So this is a relationship of the fungal hyphae inside the root cells themselves. And so here we see an actual micrograph of actual plant cells. These would be parenchyma cells, which are the storage cells in the roots of a carrot or potato or any plant root. Here it is diagrammatically. So here's our cell. And the fungal hyphae are on the outside, and then they go through the cell, and then they proliferate. So they form this highly branched pattern. And we call this branching an arbuscule, like an arbor, like a tree shape. Okay, so this branching pattern is like greatly increasing surface area for exchange. So the fungus is bringing in water, it's bringing in minerals, it's conferring some disease resistance, some secondary metabolites to prevent infection, and the tree is giving it its sugar. Okay, so we have the arbuscles. So these fungi used to be called VAM fungi, vesicular arbuscular mycorrhizal fungi. We now call them just AM fungi for arbuscular mycorrhizae. And the vesicle part was the V. So they also will produce these very large, round, swollen tips of the hyphae, and just is a storage unit for lipids and waste products. But when they want to reproduce, they'll actually produce a very thick-walled, very large spore. Actually, can be up to like 25, 30 microns externally. And so you can sieve soil with very fine soil sieves and actually see these glomospores with the naked eye. And you can actually use a fine forceps and pull them out and grow them. So the cool part about these fungi is that their role is so integral to metabolism of plants and to soils that they haven't evolved. I mean, the relationship has stayed very, very constant. So there are only a couple of genera and species of these fungi, and they are ubiquitous still. So they've done a very good job. They've done it so well that they haven't had to, the natural selection has not worked on them to select for mutated individuals. They're just very, very successful, evolutionary successful organisms. So it was a woman, Sarah Wright, who discovered this molecule, glomalin. Here it is, glomalin. Some people say glomalin. that is produced by these AM fungi. And this molecule is very, very sticky. It's a carbohydrate, but it's also got some protein. So it's really a glycoprotein, a sugar protein. And it's very, very sticky, and it's very, very durable. In fact, it can last 40 to 50 years after the fungus has died or senesced in the soil. So this molecule is the glue of soil aggregation. So here we see on the right what the soil looks like before the glomalin is extracted. And then we see what just the glomalin itself looks like, very, very deep, giving the soil the dark chocolatey color. And then we see once the glomalin's removed, just bare mineral soil, which would be a silt, sand, or clay. So the properties of healthy soil come from this molecule produced by these ubiquitous endomycorrhizal fungi. Pretty darn cool. This is seeing glomalin stained with fluorescent staining. Okay, so I've mentioned this, the glycoprotein. It has a lot of asparagine, the amino acid asparagine, and some metal ions chelated in there. And it's beneficial to most of our crops. Okay, so the only crops that we have, or veggies in the garden that we have that don't have these fungi, are the Brassicaceae. So the brassicas don't form these relationships with these fungi. So when I get to the gardening part, you're allowed to till your brassica rows, but not your other rows. Okay. So we find them in all soils. They produce in large amounts. Then they're just really tough. They don't dissolve in water. They're resistant to decay, this molecule. And so they protect the hyphae from nutrient loss. They act as like a sealant. They glue the soil particles together to form soil aggregates. And once we form soil aggregates, we leave void spaces for water to be held, for air to move, for invertebrates to navigate. And then they're just very stabilizing. So once you restore the soil sponge through these fungi, you mitigate erosion by wind and water. You mitigate drought, you mitigate fire, just because it holds the water so well. So for every gram of carbon that we add back to the soil, we can hold 8 grams of water. So that's a nice little thing to remember. For every gram of carbon we have, we can hold 8 grams of water. And there are instances where the Paradise, the campfire in Paradise, California, that swept down that canyon, within matter of hours and burned everything except one person's property that had an incredibly intact soil sponge. The fire went around them. And there are just examples of when Irene came through, hit Vermont more than us in New York, but some of you may be from Vermont. You could see the difference between the forest, which didn't erode, and the roads, which did, right? So the difference is the health of the soil there. Okay, so it's glomalin, really, really important. There's new research out of Rutgers that's mind-blowing. It's a process now called rhizophagy, or plant roots eating bacteria and spitting them back out again. That's worth another whole lecture, but here's just a scanning electron micrograph of root tips just coated in bacteria. And this process, I'm trying to remember the researcher's name, but I'm not, is where at the tips of the roots, the roots will actually take in the bacteria will dump their load of nitrogen and phosphorus and selenium and sulfur and potassium, and they'll lose their cell wall, they'll dump their load, they'll get moved back and then spit out a root hair, reform a cell wall back in the soil and collect the nutrients again, and just do it. It's like a little conveyor belt going on in the root tips with bacteria. Mind-blowing. I told you we know nothing. Okay, so those are the endomycorrhizal fungi are mycorrhizal with herbaceous things and sugar maples. So it took me a while as a mushroom forager not to go into sugar maple forests to look for big fleshy fungi for the dinner table, because there aren't very many. So they're mostly microscopic mycorrhizal fungi with sugar maples. However, all of our other trees of oaks and beeches and birches and hornbeams and all of these are ectomycorrhizal, as well as all of our ericaceous shrubs and viburnums and all the other erication, or all the other shrubbery that's in the forest. So ectomycorrhizae, ecto means outside, mycorrhiza, the fungus outside, the myco-root relationship. And these we actually can see with our naked eye. These are just 4 examples of different forms of them. Easy to identify. So I'm going to start at the bottom. So here we have the little Y-shaped forms that are dichotomously branched into little Ys. Here we have those that are dendritic. So they have a central stem and then little side branches like a tree. And then we have these that are highly branched, we call coralloid mycorrhizae. And I don't know what we're going to call these except fuzzy. So it's sort of a mixture of some of these forms. But you can see they're highly diverse. And what they're on is the short roots near the surface of the soil, right? So most of what's going on is in the top 10 to 15 centimeters of the soil. And the other really interesting research that came out of Cornell probably 20 years ago, maybe 25 years ago, was how trees use their taproot, their deep-rooted taproot, to actually pump water at night from the groundwater up and then release it into their short root system near the surface to water their mycorrhizal fungi.
Wow.
So I've seen this multiple times. My study plot in Northern California was the first year I had a very wet year, which I got all the fungi, decomposers and mycorrhizals. The second year I just got mycorrhizals and I'm like, what's going on? It's really dry. This eventually I got to reflect back and say, oh, it was really dry. And so only the mycorrhizal fungi are fruiting because the taproots are watering them. And then I saw this in Maine happen too.
Yeah. Sue, there was a question, do fungi live inside us like they do with plants?
Yeah. So yeasts are single-celled fungi. So we all have yeasts on our skin and in our moist places. And they've actually found yeast in breast milk and nursing mothers. Again, I don't think we have like mycelia hanging around unless we have ringworm or another fungal infection. But yeast for sure. Yeah. So at least as far as we know so far. Maybe we haven't looked for these things, you know. So anyway, we can see these with our naked eye if we go digging in the subsurface layers of the forest. This research was done by David Reed out of Britain, probably 30 to 40 years ago. And he really wanted to see what was going on in this interface exchange of mycorrhizae with pine. So he set up a plexiglass arena. He had multiple pine seedlings, these little short pine seedlings here in the arena. And he radioactively labored the phosphorus and followed where it went, like injected it into the soil and then followed where it went. So here we have the pine. Here, the brown parts of the pine roots going down here. So we have like maybe 3 or 4 of those. And the yellow are those ectomycorrhizal formations. So in ectomycorrhizae, you have a very prominent sheath called the mantle, which is several cell layers thick of fungal hyphae. And then the hyphae only penetrate between the cortical cells in the plant. So they don't go into the cells, they just go between to get their sugars in exchange. And so here's the actual connection, these yellow globs here. And then all of this white is just mycelium. So you can see that the plant roots go this far, and then the fungus extends the surface area in 3 dimensions, right, out from the root. So just vastly increasing surface area for absorption. So what Reed discovered, what we would have predicted in Darwin's model would have been this seedling competing with the one here and the one over here, right, for nutrients, water, space, light. Reed showed that the phosphorus was stored in the mycorrhizae here. And then if this plant needed a little bit more, it was translocated through the network over to the other plant. So all this phosphorus he showed was being mutually shared among all the little pine seedlings in this plexiglass arena. Mind-blowing stuff, mind-blowing stuff. And then of course, recently, if you've heard of Suzanne Simard's book this year, Finding the Mother Tree, she actually followed these connections and demonstrated that Douglas fir shares with birch, when the Douglas fir is, has leaves and photosynthesizing in the winter, and the birch has no leaves. And in the summer, when the birch outshades the little Douglas firs, it reverses. And then the mother trees are actually feeding their own kin, but also other species around them. So it's one giant share fest. It's just full of love. It's really like, we need to change the world to this paradigm instead, really fast. Okay, so now we get into soil practices, and I'm just checking my time. Okay. So I arrived in Georgetown, Maine to some friends for a little visit, and they had just finished rototilling this vegetable garden. And I went, oh man, I'm too late. So it looks lovely. The soil's fluffy. It feels aerated. It's really easy to plant in. It's neat and orderly, and there's no weeds. And oh, doesn't it look wonderful? And they've got their deer fence up, and the the brown, what is it? The last invitation, the brown-tailed moths, the caterpillars are just dripping from these oak trees. I mean, every fence post is just covered in these caterpillars. So this is what we don't want to do anymore. And yes, it has a short-term beneficial effect for aerating the soil, but then the minute you do this and then you go walk in there to plant something, you know exactly what happens. It goes shoop, right? And the soil gets very compressed. Okay, so what we want to do instead is we can take it in baby steps. So here's a local cornfield on Route 22 where we now, thank God, are just cutting off the corn and leaving a good 18 inches of stalk. And then the stubble is getting left behind as mulch, right? So it's a cover. So this is like better than tilling to bare soil. What's even better is to use a cover crop and plant between the corn. This is a ryegrass that's planted between the corn, timed so that after the corn is harvested, the rye still has plenty of time to grow in September, October, November. And then in the spring, it will be rolled flat, and then the corn will be planted directly into the rye. And then what I've heard from our local farmer who's doing this is that other people's corn looks better at the beginning of the summer because of their fertilizer inputs. But by August, when his rye is fully decomposed, there's this huge nutrient flush to his corn in mid-August that the other farmers don't have. And so his corn goes zoop, and look beautiful by mid-August. This is just an example Dee Dee Pershouse used when I was studying with her of this family ranch in Africa, where you can see the differences on both sides of this fence from, animals not being on this land and just not be able to support very many animals. And you've just got baked soils. The temperature of the soil is way higher than the temperature of this soil. You've got so much compaction and baking of the surface that no water is going to penetrate when it does rain. So there's no infiltration. And over here, where animals are used in a very controlled fashion, they're releasing the microbes, they're stirring up the soil, allowing for little divots for water retention and seed collection, and fertilizing at the same time. And you just have this very healthy grassland effect. So this is just one example of how to use soil, healthy soil practices. And so when we look at our own gardens, ultimately we're going to want some little gadget that measures soil carbon, but it doesn't exist yet. I'm working with some researchers out of Hudson Carbon down in Hudson, New York. I'm actually going to meet with them tomorrow. And they're working very hard on how to quantify what's going on in the soil. How do we measure the carbon that's being stored and compare, right, to know that we're doing the right thing. But there are some easy techniques I'm going to show you now where you can test yourself to see if it's better. Like, you won't get an actual measurement, but you can say if it's an improvement or not. So this is a soil simulator by Benton James, where you've got 5 arenas here. You've got bare soil, you've got a teeny little bit of cover, a little bit more cover, a little bit more mulch, and then you've got living plants and living roots. And so we add the same amount of rainfall to all of these, and we see that on the one that's bare soil, most of it runs off the surface into this jar, and we have in the bottom jar nothing infiltrated, like nothing is getting through. The soil is a crust, it's baked, it's going to have the water run off. Here we get mostly runoff, just a teeny bit infiltrating, just a little bit saved by this plant matter. And as you see that it progresses in inverse relationship here. So more coming through, less running off, to where we get over to the living plants and there's no runoff and it's all running through. And guess what? It's clear. So we're not losing soil particles. We're not losing nutrients. We're not losing sediment. Okay, so it's a simple little demonstration, and you can do this with little rings, just pounding them into your soil and measuring how long it takes for the water. There are certain sizes and volumes you have to use to make it standardized, but you can measure how fast the water infiltrates your soil. So I've been in farms locally where it's taken 45 minutes. And then I was on a Stonyfield yogurt dairy farm where the pasture hasn't been touched in 80 years by this family. And literally the water went through in 45 seconds. And we dug up that soil and it was chocolate cake. So what you want is very crumbly, dark chocolate cake. That's the premier soil test. So here are the infiltration rings that you can pound in and do that previous test I mentioned. And the slake test is another simple one. So this is just to measure how aggregated your soil is, how much glomalin is in your soil, how well it's being held together. So you fill up 2 jars first with water, and you take a little window screen or a little cheesecloth and just, suspend it with a rubber band around at the top. And then you take a measuring cup and measure out the same amount of soil from 2 spots, and then just gently place it into the cheesecloth and just see what happens. So if your soil does not have a lot of life in it, it's going to fall apart. And if it's got lovely microbial life in it and lots of glomalin, it won't. So this is simple things that, we can do to measure change, right? So if we want to start implementing these practices in our own gardens and our own crops, we can start looking at it now. We can also do spot testing to see if there are different parts of the field that act differently, which there will be.
Sue,
Yeah.
Is the glomalin the same thing as humus?
No.
Okay.
No. So humus is just going to be smaller particles of plants and excrement and, so decomposition happens not just by fungi, right? So we have the shredders, we have all these invertebrates and arthropods that break it down into smaller and smaller chunks, right? And then the fungi work on it. And actually worms do the final piece of passing it through their digestive tract and excreting it really to get all the way decomposed. But no, it's not the same. And I can't tell you, maybe somebody on the call can, chemically what humus is. I mean, it's going to be a combination of a lot of things. It's not just going to be glomalin. And there could be glomalin in it, but that tends to be really just affiliated with the roots, right? So, and the hyphal extensions from the roots. So, so if we want to restore, I started doing this after taking courses from Paul Stamets 20 something years ago, where I was just trying to restore forest trails and logging roads and things at Skidmore where there have been disturbances from either construction or mountain bikes. So we're doing a oyster mushroom wood chip installation here in the background. But as I said at the beginning, it's just a mere 2% of increasing the soil carbon would be this huge offset of greenhouse gases. And we have to stop the emissions from going up. But Walter Yene, the second author here from Australia, he started Healthy Soils Australia. He's got awesome 2-hour TED Talks if you really want to get going on this stuff. He's just a delight to listen to. But he is the most brilliant biochemo physicist. He gets all of it. He gets biology, chemistry, physics, and he gets the water cycle. And so really, if you listen to Walter, it's not the carbon that's out of whack, it's the water cycle that's out of whack. Our planet is warming because of the water cycle interruption. But water is too hard to model versus one man's data in Mauna Loa, Hawaii, right, in the top of the mountain from 1958. Charles Keeling monitored carbon dioxide concentrations in the atmosphere 1 mile above the ocean And it's his one person's data set that we have based all of our, I know David Higby's on this call. I'm talking to you, David, all of the climate change policy on erroneously, right? So Yene says fossil fuel emissions only contribute 5% of the CO2 in the atmosphere. Far more comes from bare agricultural soils and then agricultural feedlots too. But so because we can correlate the rise in parts per million of CO2 on Keeling's curve, it's called the Keeling curve, over time since 1958, we can go, oh yeah, well, that's obviously the cause of warming of the planet. And in fact, it's the lack of water in the soil that keeps us cool. It's deforestation that keeps us cool. You know, the deserts of the Sahara, all of the deserts of the planet used to be lush vegetation, right? Civilizations collapse because all civilizations have collapsed because they mine the life out of the soils. And we're headed there. We are headed there. If we don't turn it around, we are headed to the collapse. You know, all of the historic civilizations, the great African civilizations first. And so, yeah, so it's cooling the planet through holding on to water. So adding carbon to the soil holds on to 8 grams of water. So that's where we start. That's where we start. So replanting and getting the carbon and not disturbing is where we go. So we have, I've already talked about what Peter Donovan started the soil carbon initiative, I think. He did a lot of early teaching about this from his travel bus, a school bus he converted. So the soil holds more than twice the amount of the atmosphere and the plants above land combined. That's just really important. And then we are going over number 3 multiple times, and that the mycorrhizal fungi play this key role in glomalin storage in this durable, hardy, long-lived molecule that really is the key. That really is the key. And so when we plow, when we till our gardens and break it up, we're destroying it. Okay. So I promised you, well, one more thing on healthy soil principles, then we'll get to our gardens and growing fungi. So this is the thing. It's not that hard. Stop tilling. Keep your soils covered with mulch and cover crops. So well, Janet knows this. Janet's been there too. One of the ASA farmers down in Johnsonville is Otter Creek Farm, and in 3 years, they have converted their farm to regenerative principles, and they have 7 crops in their cornfield, 7. And in their pasture, I think she said they're up to 18 species of forbs and grasses in their species. All of their Red Angus raised beef and their pigs and their chickens are fed nothing but the grass. There's no grains, there's no extra feed in there. So there's no cost, there's no fertilizer cost, there's no pesticide cost. Their fields are gorgeous, their animals are beautiful, and I can taste the love in the pork. Okay, so the more we increase biodiversity above ground with cover cropping, the more we increase the microbial populations below ground. That's just common sense. And then the thing that's not as intuitive, but is really essential is, we are used to feedlots and huge concentrations of livestock, which is not healthy, right? Because they're not on the land, they're in a feedlot. But if we use the animals as evolution has done, right? Grasslands exist because of herbivores. That's the only reason they're around, right? So our pastures and our fields exist because of grazing animals. And so the grazing animals have evolved with these plants. And so we do need them. And so if we graze them this is in Ireland, the bottom picture, a linear fashion. I've seen radial spokes of a wheel fashion, or just, paddocking off, a couple acres at a time, and move the animals through either every day or every couple of days or every couple of hours, depending on the amount of space you have. You give the plants time to recover so that by 8 days later, 20 days later, however much your space is, down at Otter Creek Farm, they have a 20-day rotation where they take their beef. It takes their beef 20 days to get back to the starting point again. By that time, the plants have recovered. It's self-sustaining. It's really low input agriculture. really low input agriculture. And then it just builds the microbial population. Okay, we're going to skip this. This is a whole talk. This is John Kemp's work. His organization is called Elemental Ecosystems. No, that's not right. That's Zach Weiss. John Kemp is an Amish farmer in the Midwest. He's in Montana, and Eco something, great organization. Anyway, this whole plant health pyramid translates directly to human health, and it's all about making photosynthesis more efficient from 20 to 30% up to 60%, and then we solve all these other problems. That's a whole nother lecture. If you want that one, I'll do that one later. So here's back to that initial slide of bare, compacted, runoff, erosion, rainfall isn't affected because it doesn't get into the earth. The soil temperatures at the surface, or the temperatures of the surface are huge. The heat is lost, greenhouse effect, dust, smoke, and then the very ideal soil sponge, lots of water, plenty of infiltration, lots of microbial growth, bacteria coming out of the leaves, cooling, and then creation of clouds. So we have this biotic pump on the planet working. Okay, so the last little bit of the talk is going to be just adding some fungi to your gardens, not just caring and maintaining the mycorrhizal fungi in the soil, but you can do a lot with growing some edible and medicinal mushrooms in your gardens as well. So this is Paul Stamets, when I went out and studied with him in 2006. And this is called cardboard culture. So you simply soak the cardboard in for like 10 minutes in some warm water, pull it apart to where the corrugations show, and then you start planting just little tidbits of stem butt. Okay, so we'll look at what stem butt looks like. Oops, I'll show you in a minute. Another way to do it is to grow on birch dowels that you're going to plug a log. So you can plug oyster mushrooms in a log, you can plug maitake mushrooms in a log, you can plug shiitake mushrooms in a log. And you can either do this yourself by getting some grooved, they're usually 3/4 inch long and 5/16 inch wide birch dowels, and just soaking them and then pressure cooking them to sterilize them, and inoculating yourself with just some oyster mushrooms you find in the woods, and just the base of the oyster mushroom, and breaking the tissue up in a plastic bag and letting it go. Here's some oyster mushroom growing on hardwood chips, same kind of thing. You can start with the cardboard culture, oyster mushrooms and cardboard culture, and then transplant the cardboard culture to the wood chip bin, and then bigger box, and eventually you go out into the land and move it out into the land. Shiitake log cultivation, I've led a bunch of workshops with pretty crappy wood, but if you have a wood lot, 4 to 6 inch diameter logs, 3 to 4 feet long of hardwood, and poplar works. Oak is the best. Maple is okay. Birch is okay. And you drill 5/16 inch bit holes every 2 inches down your log, and the rows are 2 inches apart, so you're alternating. You just tap in the colonized birch dowel that you saw in the last picture, and then you seal those holes, and you seal the ends of the logs with a wax. Waxes are expensive, so there's cheese waxes, and there's beeswaxes, and then there's paraffin, and there's the stuff I order in 50-pound blocks is a combination of all those things. But you're just trying to seal the moisture into the log. And then you want to keep these logs in a very shady place, but this is what you get, and you can get it for 5 years and then start over again. And you know what these sell for in the grocery store. You can get up to 55 pounds per log. So back to my favorite mushroom to grow in the garden, which is the wine cap or giant straw mushroom. So these grow naturally. And the first one I found actually and brought into culture was in a cornfield. They grow at the base of the corn stalk. So you can look for them there. They are called wine caps because of this beautiful red wine color of the cap. There is a yellow version here in Washington County that's golden yellow on the cap. And they also bleach out in the sun. So this is this over here. So the sun will bleach them out. at the base, so this is a decomposer fungus. It's also a sun-loving one. What's the word I'm missing? Something file. I can't get the word out of my brain right now. But it needs sunlight to grow, and it also needs bacteria to grow. So these guys actually feed on bacteria, as well as breaking down the carbon in the cardboard or the wood chips. So at the base of these, you'll find these white rhizomorphs that look like little root structures coming out the bottom. And each one of these rhizomorphs is a aggregation of about 20 to 30 of those microscopic hyphal cells. So you can actually see it with your naked eye. Stamets discovered that the combination of the rhizomorph and the stem butt, so this whitish soil covered area, is regenerative on its own. It's like a lizard tail that can regenerate, or a starfish arm that can regenerate. So there's no sterile anything. I just use my bare hands when I find these, and just take this thing and break it up into 5 little pieces and plant it like a 5 of spades card, just on the cardboard. And you put it just in a little Tupperware container, put the lid on, and I burp it every day just to give it some air. And in about 4 days, you're going to see this. So here's a little crummy piece that I ripped out of the bottom of that mushroom. It's not looking so good itself, but it's growing from there. And you let this colonize the cardboard for about a week, plus or minus a couple days. And then you're ready to transplant your cardboard into a box of soaked hardwood chips. So I just use a cardboard box, put in hardwood chips in the bottom, wet them, soak them thoroughly, plant my cardboard, put in more hardwood chips at the top, fold the box, and I usually just stick it outside under the spruce trees. And in a good year, it's covered with snow all winter. And then I forget about it until I'm in the garden in May and I go, oh yeah, I had that box. And then I go look and it's just beautifully colonized and I'm ready to outplant it into the berry patch. So to outplant, you have to give it a head start by putting down wet cardboard to keep the soil microbes from competing with it. And then after the cardboard, you're going to lay down more wood chips. You're going to lay down your colonized wood chips with the fungus, and then more wood chips and maybe a little straw. And then you're going to have to feed your patch fall and spring. So if you start your patch in the spring, you're going to have to add more wood chips in the fall, add more wood chips in the spring, and you can keep that patch going for about 5 years as well. The first year I did this, I was picking 20 mushrooms every morning and 20 mushrooms every night in an area that was only like 3 feet by 15 feet long in my berry patch. And I had to pick twice a day because you can see here, the deer came along, and these are deer bites. There's a deer lip there and a deer lip there. So the deer came along, and then the little mice came along, took some nibbles. And so that's why I was picking twice a day. And you do have to recognize this fungus because other things can fruit in there too. And so I could never send my husband out to do this because he couldn't distinguish. So it's very easy with this fungus. You've got the color. It's the size, fully grown, it's the size of a portobello. You've got this beautiful lilac gray color of the gills. Here you see it over here, more mature, but it's a very pale gray, and then it gets deeper. And then you've got this very prominent ring around the stalk called an annulus, and it's a double ring. So that means that it's on this one that hasn't opened up yet, where the ring is still attached to the cap. There's like a broken up yellowish tissue here. You can see it on the bottom of the ring, and then the top of the ring is going to be white. So it's called a double annulus. So it's got 2 kinds of tissues going on. That's pretty unique to this fungus. And then the stem butt has to look like this with these rhizomorphs coming off of it. And if you wanted to make double sure, you'd do a spore print by cutting a cap off and putting it on paper. And the spore print will be a dark purple brown. And then you can always send pictures to Sue and she will confirm for you. I'm happy to do that. But this, I know some other people up in Salem that are using this. You can order this mushroom through a number of supply companies. I use North Spore in Westbrook, Maine to get my spawn if I can't get it from the wild. But giant straw mushroom is awesome. And this is another one you can grow easily in your garden. The very first year I moved here, I didn't have any mulch, so I shredded newspaper between the rows of vegetables. And I got this fungus all over the newspaper, just loved it. And this can be confused with some brown spored mushrooms, so you have to be a little bit careful, but these are wood bluets. They love cold weather. They will fruit all the way into November. I even had some in my yard this year in December. If they start out this beautiful purple color, and they love, here's a little hemlock cone. See right here? So these were fruiting under hemlock. They love pine, they love hemlock, and mine were under a yew tree that we cut down this year at the base of the yew tree in the grass. So the margin of the cap is enrolled here, which means it curves under. The gill attachment is a little notch gill. Beautiful, it's just beautiful purple all around, purple stem, purple gills. It will fade to a beige color, and it's a very waxy cap. And the spore print will be a pale lilac, but it's a very delicious fungus and you can easily cultivate it. And then of course, oyster mushrooms in stumps. So when we were doing some trimming, I said to George, oh, leave me 4 feet, please, leave me 4 feet. And don't get rid of stumps. Don't have people come in and grind your stumps. Like that is fungus food. That is really good fungus food. Plus I have wonderful pileated woodpeckers in my stumps, just inside of them. So this was my first year after inoculating the little dowel plugs into this stump. And this was like the 5th year. So you can see how much it colonized. This was a butternut, actually, very wet wood, how much it colonized. And then that's just edible. So we could go to medicinals and have another whole talk. But I think we're getting pretty late on time here. And I don't want people to, but we have chaga locally. We have hen of the woods, highly medicinal. We have birch polypore. We've got the lacquer conch on hemlock. Shiitake is very medicinal, also vitamin D. We've got cordyceps fungi, and we've got Fomes fomentarius. So all these polypores have, they're persistent. So they have to have a chemical warfare to keep things from destroying them. So the secondary chemistry that they've got going on is where the medicine occurs. And then turkey tails are being used for breast cancer research. Lion's mane is being used for Alzheimer's and nerve regeneration. You can buy this at the farmer's market, the lion's mane from Jeff. And then, of course, psilocybin mushrooms. It's so fascinating, but the last 30 years coming out of Johns Hopkins medical team and researchers, I've met a lot of those people, and the effectiveness of controlled doses of psilocybin administration for treating trauma, anxiety, depression, and cancer. Anxiety is incredibly effective. Like we're talking one session, one dose, and people are healing from deep trauma. So Oregon's made them legal. They're still a controlled substance, but lots to watch there. In addition to all of the Mesoamerican sacred use of fungus. This is on Paul Stamets' website. Just, here are some of the species of fungi across the top, and here's, antioxidant support, blood sugar support, breathing, digestion, energy, stamina, heart support, immune response, liver detox, memory and clarity, nerve support, performance recovery. So you can see where some of these, like reishi mushrooms, down the line, whoops, the whole thing. Just like reishi is a immune modulator. So if your immune system is overactive, it'll pull it down. If it's underactive, it'll pull it up. And a very long 4,000-year history of this fungus in Asia and Japan and China. This is a fungus you can take every day. The one that's driving me crazy is chaga, where people are using it every day and cause coffee supplements, and chaga is too powerful. In my experience, it's just way more powerful. You use it when your body needs help, not every day. So, here we are with, this was before I learned about soil regeneration, of just using mushroom tiles from Ecovative as my mulch, and they would decompose in 4 months, and my soil is just so good because of all that inputs. But now I try not to disturb it at all, and I keep it covered all winter. I keep my plants in there. I don't pull them up in the fall. I just let them be there over the winter, and I'm now just trying to barely scratch the surface to put the seeds in. And you can do experiments in your own garden. Like you can try a little plot where you do till it and move it around, and then don't do it and see the difference.
Sue, can I ask some questions here?
Yeah.
So when we talk about tilling, we're not just talking about rototilling, right?
Correct. We're talking about using a shovel
Or a garden fork.
a cultivator.
Mhm,
Yeah. I mean, I've always turned my garden by hand with my shovel.
mhm.
And then added my compost in too. Like last year, I did not add in my compost, and then those jumping worms showed up, and I was really glad I didn't. But yeah, I when I planted my garlic this fall, I did mix in the compost. So I did till that in.
People who have gardens that are very weedy, I mean, even pulling weeds can be a form of tilling.
Yes. And so what I'm trying to do is just cut them off.
Mhm.
I have a big burdock plant in the middle of my garden. And so the burdock root is like awesome. And so, I mean, it's a great conduit. So when it gets to be too much growth on top, I just cut it back, and then it grows again, and I cut it back.
So
Yeah.
cutting off weeds and then mulching
Yeah.
with weeds, mulching
Yeah.
with other things.
Yeah, I mean, you have to watch the seeds, right? But you know, the weed seeds, so you want to do it before they go to seed if you can.
Mhm. Yeah.
Yeah. I mean, it's a whole different way of trying to think about it. And
It's a whole different look. People have to get used to a different look.
absolutely. 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 And then the benefit comes in really healthy looking plants and fewer pests on your plants.
Mhm.
You just have to wait for that.
Sue, there were a couple of questions. What do you think about the whole jumping worm situation? They somehow got imported into my garden. I tried to use denial for a while to see if I could make them go away, but it didn't work.
Yeah, I don't have them yet, so I'm not real. I know Bliss, you've described to me that they turn the soil to like coffee grounds, and really chew up the organic matter. I don't really know enough about what to do with them.
It was really interesting in the talk that I heard about them. Someone asked, why don't they have them in Japan? I mean, why aren't they a problem in Japan? And they said it's because they don't have the kind of maple leaves that we have. I mean, I was interested that you talked about maple leaves, but maple leaves are so nutritious and easily decomposed, easily eaten by the jumping worms, so they can proliferate. And in Japan, they have more evergreens. They have evergreen oaks as well as the
Okay.
conifers. And it just isn't as delicious a leaf litter.
Okay, so I know that when we do ecological studies and try to measure the earthworm population in the soil, you make a solution of mustard seeds,
Mhm.
and you pour the mustard seed water on the soil, and all the worms will come up to the surface. So I suppose that's one way to see if you have them in the first place.
Well, I think where I got them was somebody brought me some composted leaves from the village,
Mhm.
and that's where they came from. I'm on a listserv for a Vermont vegetable growers thing, and I saw that there's going to be a webinar on jumping worms, I think later this month. So if I glean any great information, I'll try to pass it along through Bliss to the garden group.
Yeah.
I did the whole blog, and it was very painful for people to read.
Oh, okay. I should check out your blog then.
A couple weeks ago.
Yeah. Yeah. It seems like it's always something, right?
Right.
Yeah.
Well, it could make the 2% gain a little more challenging.
Yeah, right. Until we find some wonderful use for jumping worms.
Yeah.
Right.
Well, plant your perennials, just keep adding the organic matter, and we can stay ahead of them. You can still have healthy plants. You can have healthy plants and jumping worms.
And, okay. Yeah.
Does anyone else have any questions for Sue right now? So we did record this, and a few people had requested they couldn't make it this evening and asked if we could send it. So I guess with your permission, Sue, can we share this, or
Yes.
how do you?
Yeah, absolutely. Yeah.
Okay, great.
And I have reference here at the ends of books that I've read that are wonderful, and then the organization's working on this, TED Talks and films, and then these are some actual primary literature articles.
Great.
So yeah, that'll all be,
Okay.
yeah.
Also, I want to share that as part of some of the outreach programming ASA is doing, earlier or last fall, I guess, Sue and I had talked about doing a program in someone's garden or a small farm. We haven't come up with a location yet, but where we will look at how to take a soil test, how to interpret a soil test. Maybe we could do we could show how to do an infiltration test, and then talk a little bit more about different cover crops, and just more of a hands-on look at building soil health in your garden or small farm.
And I want to add to that too, Janet, that I have a microscope, and
Right.
we can actually sample some roots and stain for these AM fungi and see if they're there.
That would be very cool.
Mhm.
Good. Well, if there aren't any other questions, I want to say thanks so much to Sue. This was really great information and very inspiring. And as someone else said, this is going to take a little review. It was a lot of information to digest.
Sorry about that.
We need the help of a fungi, I guess, to help digest it. So thanks again, and we'll let all of you know who registered when we're planning the soil health talk in the spring or early summer.
Yeah. And I highly recommend watching some of these films, because that is the real experts talking about this. And the more times you hear it, the more it becomes ingrained and easier to understand. I mean, I had to read all these books to be able to get here, and I know this stuff. I mean, I know the plants and I know the fungi. So it's still taken me a lot of study and review, and I've been on so many webinars, I can't even count them all, on just hearing it over and over and over and over again to really be able to talk about it myself. Yeah.
Great.
Yeah.
Well, thanks again, Sue. Really appreciate your time and expertise. And thanks, Bliss, for suggesting this. And thanks to all of you for joining us. And keep an eye out on our website for other programs. Thanks and good night.
Good night, everyone.
Janet, are you going to be emailing that to everyone?
Yes, we will try to do that. I'll have to figure out how big it is and what's the best way to send it. But we'll work on that, okay?
Thank you very much.
You're welcome.
Great show, great talk.
That's really very interesting.